So I think I would start then,
Yep.
right?
Yep, go ahead, Roland. We're with you.
So welcome to our Phenomics webinar. I think having a global distribution of participants, I can say good morning, good evening, good afternoon. It's a pleasure to have so many participants here from all over the world. And we have today a very highly interesting topic, on namely root phenotyping, given by Malcolm Bennett. But first, let me briefly introduce you what's the background of this webinar and the involved projects that organize that. Very briefly, the webinar, the goal is to have an exchange platform for the community, and basically also partly to compensate for the COVID-19 related limitations that many workshops, meetings were cancelled, and we wanted to compensate for that and provide a platform for exchange. We have two different formats for the webinar, namely a single talk like today and thematic sessions where we specifically address a specific topic and have multiple sessions. And the webinar is pretty open, like at the conference, there is a submission of abstracts, and we welcome contributions all over any career levels and stages from anyone interested in plant phenotyping and who can contribute to plant phenotyping. Please share your research with us. It's interested for anyone to share it in those times. The webinars take every second Friday at 14 Central European time. Basically, so already advertising for the next webinar. In 2 weeks, we will have Nadia Al-Tamimi talking about salinity tolerance. So very briefly, the backgrounds, we have 3 projects that are the organizers. It's IPPN, the International Plant Phenotyping Network, which looks at integrated regionally, globally fragmented activities in plant phenotyping, but also enable and facilitate exchange of knowledge, information, expertise between any experts involved in plant phenotyping. IPPN is open for any participants to join and become members, so please feel free to join and become member of IPPN and support activities like this webinar. Additionally, EPPN 2020, this is also a community-driven project where we have one of the key elements in EPPN. It's access to 20 to 31 facilities across Europe. Currently, we have the 6th and final call, which is open. The deadline is in October, end of October. So the application is based on a very simple application procedure. Once your proposal is accepted, full costs are covered, including traveling to the facilities. And we obviously 20% of the projects can be from non-European labs. This is an opportunity which provided already a very good output. We had applications so far from 124 projects from 34 different countries, so it's a wide distribution, it's actually also across the globe. And we have substantially more than 70 publications right now, so there is not only that we provide the opportunity to do experiments, but it's there is a substantial output coming out of that. Additionally, the goal of emphasis is to provide long-term perspective, long-term development in providing access and other services and resources related to plant phenotyping for users from academia and industry to use those. The goal of emphasis is to develop pan-European infrastructure, provide access to this infrastructure, enable genotyping of performance in diverse environments, share knowledge and novel technologies within the partners involved in this project. And there are 5 different infrastructure categories related to controlled environment, fields, as well as data and modeling. And around those categories, there will be a set of services, resources, and access models developed that will be provided on a long-term basis. Additionally, the webinar is also broadcasted in China via our partner platform, AgriForum, and it's supported also by the University of Nebraska. It's the technical support, which we really very appreciate. Some simple rules on how we would proceed with this presentation. So during the presentations, please feel free to put questions into the chat at any time you wish. And there will be 3 different sections of the presentations. And after every section, there will be time to basically raise those questions. So please put those questions into the chat and we will read these questions and Malcolm would address your questions. Additionally, if we have some time at the end, we can also, you could also raise a hand and you can also basically orally ask your questions. So moving now to Malcolm, our speaker today. So Malcolm was throughout his career highly interested in root biology. His major stations in the career was the University of Warwick, where he did his undergrad, PhD. Then he went to University of Arizona as a NATO CERC fellow, came back to University of Warwick, spent there more or less the 90s. In 1999, he moved to University of Nottingham, where he was active in many different positions and did a substantial development, and not only relevant for Nottingham, but for root research, I would say, across the globe. Malcolm is has more than 200 research papers and is ranked among the 1% mostly highly cited animal and plant biologists, received several awards, honors, including the Royal Society Wolfson Research Fellowship. He's an elected member of EMBO, honorary degree from University Catholic Leuven. He's elected fellow of the prestigious Royal Society. So he makes the plant science, the work that we do, visible way beyond the plant community. And we are very pleased today to welcome Malcolm and talk about advances in root phenotyping. And was mentioned, there will be 3 different parts: field, controlled environment, and new directions in root phenotyping. And there will be time for questions after each part. So Malcolm, please, the stage is yours, and we are very much looking forward to your presentation.
Thank you very much, Roland. Thank you very much, Jennifer. And thank you for everyone who's joined this afternoon. It's a great pleasure to join you. I'm going to share my screen. Okay, and so my question is, can you see this?
Yes, yes, we can, Malcolm.
Fantastic. Thank you, Jennifer.
Mm-hmm.
Okay, so thank you for the invitation to talk about advances in root phenotyping. As fitting with such a huge topic in which many people have contributed, I've used this as an opportunity to illustrate not just my own group's work, but also the work of collaborators and also other colleagues in the US, Asia, and also in Europe. So I'd like to, first of all, ask the question about why are we interested in root phenotyping? Well, obviously, in a very simplistic way, we've spent 10,000 years really looking at the top half of the plant and not really, I think, appreciating the importance of that the lower part of the plant, the hidden half, actually contributes. And pioneering work by the likes of Jonathan Lynch, Uga, and others are really rewriting our understanding of the importance of root biology for crop performance. So this schematic really illustrates how simple changes in the architecture of a root system can really have profound effects in terms of crop performance. So for example, the root angle, the angle of roots close to the surface in which phosphate accumulates, can really have a profound effect on the ability of roots to acquire this key nutrient. Root hairs, likewise, can also have an impact, as beautifully illustrated through the work of Jonathan Lynch's team. Branching density can also have a role. Root angle is extremely important in terms of water capture, as beautifully illustrated by Zaku Uga and his DRO1 work. So we know that the architectural properties of the root system are truly important for many of the challenges that agriculture and society face in the coming years. We want to make our agriculture more sustainable and use less fertilizers. Clearly, having a more efficient root system to forage for these nutrients is important. Likewise, developing crops in which are more climate proofed through, for example, steeper rooting to secure water supplies can be achieved in part through selecting crops on the basis of their root systems. Now, obviously, this is not simple. We're talking about assessing these traits when the roots are obviously growing within the soil systems. So these obviously places a number of practical challenges to assess these traits. So what I'd like to do today is walk through three different topic areas in which we're assessing root architecture. And the first topic is obviously assessing roots in their natural environment, going back to the fields in which they grow and assessing their traits. And this, I'll use two projects that I'm part of to illustrate some of the challenges and solutions and opportunities which result from this. The second theme will be looking at imaging root traits using controlled environment conditions in which we're able to obviously obtain far higher resolution analysis. And I'll describe some of the discoveries that our team has made as well as others. And finally, I'd like to finish by talking about several new innovations in root phenotyping, which myself and our collaborators are contributing towards. And obviously, this is not a comprehensive analysis, and we're obviously interested in hearing other comments in the discussion sessions that punctuate this. Okay, so I'd like to start by addressing the challenges of field phenotyping for root traits. And I'm going to, as I mentioned, use two projects to illustrate this and the solutions. And the first one addresses the issues related to drought stress. And this is a project which is looking at improving rice root architecture to address the pressing issue of drought stress, which threatens global rice production. As you can see, many of the rice production areas have highly variable average rainfall, and this can obviously be affected, can severely impact crop yields in these production zones. The area that this particular project I'm going to discuss focuses very much on Southeast Asia, and in particular on Thailand. The Northeast Plain, which is a major rice production area within Thailand, suffers from drought stress, and this project attempts to address the impact of rice root architecture. The project, which was led by Jonathan Lynch and Leah Band at Nottingham and Penn State, it's called RhizoRice, and this schematic that's on the slide really illustrates the opportunities that root architecture presents. So on the left-hand side, we see a typical paddy rice style root architecture characterized by varieties like IR64, in which the rice roots are shallow, relatively dense. And this project explores the opportunities that could arise from having different root angles, different lateral root densities, which are architectural traits, but it also addresses the impact of not just architectural traits, but anatomical traits. And this is an area in which Jonathan has been a real advocate and a pioneer, and I'll show some results which actually really suggest that these are very promising. So anatomical traits such as vascular xylem diameter, which has been pioneered also by breeders such as Richard Richards, is explored in this project. So in terms of the environments in which we are doing the field phenotyping, these were a combination of natural drought environments at several sites across Thailand, and also work was done by Amelia Henry also at Erie under more controlled conditions. Field phenotyping obviously is incredibly challenging and backbreaking work, and so root phenotyping is still a very physically demanding process. Techniques such as shovelomics, as is illustrated in the top left-hand corner, pioneered by Jonathan's group, allows, simply from characterizing the top part of the root, you can derive a lot of important information such as root angle, as illustrated in the lower panel, which can infer a lot about the underground root architecture. Many other techniques are also were employed in this project. For example, soil coring is a great way of obtaining information about root length density at various depths. Baskets are also very, very effective, as is illustrated through Zaki Uga's work to identify DRO1. And rhizotrons could also be employed. Clearly, once the materials were isolated, then various software approaches were developed and applied. So we took advantage of Alex Bush's DIRT software to characterize root architecture. But also WinRhizo, a commercial software, is also still a very useful software to assess the various classes of roots which you find in rice. And finally, in addition to disruptive approaches to look at architecture, we also employed a technique which is pioneered at Penn State called laser ablation tomography, which I'll talk a little bit later in the new advances section. As you can see on the right-hand side, if you can see my cursor, we measured a very large number of different traits whilst the field measurements were conducted. So this was a huge amount of work done by a brilliant team in Thailand. Nan, Jonah and Kwan led a very talented team. And I'd like to just share a couple of results with you to illustrate the importance of why we think root angle is really important. So during this process, we characterized a number of shallow and deeper rooting rice varieties, Thai rice varieties. And it was interesting to observe that under well water conditions, neither sets of varieties appeared to impact shoot dry weight under well water conditions. But when drought stress was applied, you could see an immediate benefit in terms of deeper rooting, in terms of maintaining and developing a larger shoot dry weight. And in fact, under drought stress, there was a significant reduction in the shallow rooting varieties, which was ameliorated to a large degree with deeper rooting varieties. Probably less appreciated by our community is the importance of anatomical traits. And as I mentioned, Richard Richards did some pioneering work in Australia in wheat many years ago, which looked at xylem diameter, which appeared to really contribute to this. Jonathan and others have really pushed this further, looking at traits such as parenchyma. In this case, this looked at xylem vessel diameter. And as you can see, there are varieties with large and small xylem diameter, and these were characterized in field studies. And again, under well water conditions, there wasn't any kind of yield gain or impact in terms under well water conditions, but there was a significant benefit under drought stress conditions. The smaller xylem diameter varieties performed considerably better. And this was really, again, a very significant contributor in terms of maintaining shoot dry weight. This trait is likely to reflect that smaller xylem diameter will retain more moisture in the soil and obviously eke the water source out for longer than, for example, probably less of water use efficient varieties with larger xylem vessels. So this work was led from Nottingham and Penn State by Jonathan and Leah, and we were very lucky to work with excellent colleagues in Thailand and also in Erie. And this is illustrated in these pictures at various project meetings, either in Thailand or at Nottingham. I'd like to move on to give a second example of field phenotyping. And this project, which is funded through an ARPA-E grant, a $7 million ARPA-E grant as part of the Roots program. This is led by Penn State and Jonathan at Penn State, but involves teams at Wisconsin, Georgia, and Nottingham. And the concept behind the Roots program was very far-sighted. They wanted to look at whether, if we could grow deeper roots, we could, it's been predicted we could sequester up to 2/3 of the US transportation carbon costs, and also improve uptake of nitrogen and water. To do this, we obviously require new breeding approaches to assess deeper rooting. And obviously, the team used a variety of approaches, tried and trusted, such as shovelomics. They also to look at architectural features, anatomical approaches, but also this was a very innovative project that also attempted to develop invasive as well as non-invasive approaches to assess rooting depth, for example, and identify key genes to feed into what we call a deep genes program. I'll quickly just pick out one or two examples to illustrate this work. One of the projects was, as I alluded to, root phenotyping is extremely backbreaking. And so to automate this through the use of, say, robotics is obviously a long-term goal. Paul Heinemann at Penn State has been instrumental in creating what is called the root robot system. And this system would allow a tractor-based system to essentially excavate the crowns, in this case of maize, to wash the stalk, and to essentially prepare this by cutting it in half for image analysis in chambers. Now this system is well advanced in terms of its design and application, and it's being used in the field this summer. In parallel with the robotics, we also have to have clever ways to capture the information, the architectural information. And this is beautiful work that's been done by Alex Bush's group at Georgia. They've developed a very nice image analysis approach in which they characterize using multi-angle imaging a real root. Note that it's actually cut by the root robot in half to uncover the internal structure. And you can see the real root on the left and the simulated captured image on the right, and it's breathtakingly realistic. Alex has performed some beautiful work to capture information about key traits such as whorl number, stem thickness, and lateral root densities, for example. And what's really exciting is that if you compare to the manual versus the automatic capture of these traits, you can see they're in very, very good agreement between these measurements. I'll give you one example of how we're feeding into what we call the deep genes pipeline. So this is where crown root angles have been characterized during field studies. This has been fed into genome-wide association analysis. And this has resulted in the identification of genes which actually control root angle, for example. We are very excited that this allowed us to identify a gene which encodes a kinase. And when we knock this out using revert gene validation experiments, we were very interested to see we could alter the crown root angle. So we're very excited how we've gone from field phenotyping data on a diversity panel of about 400 lines, the WIDIV population, assembled by Sean Kepler's team in Wisconsin. And when this is identified, key genes, and when we've done gene validation, we've been able to alter root angle. And through some very nice work by Hannah Schneider, who's based at Penn State, using greenhouse and field analysis, they were able to show that the change in root angle improves nitrogen capture. So deeper rooting maize varieties are able to, in both field and greenhouse studies, to improve nitrate capture under either high or low nitrogen conditions. So this is a very nice example to illustrate how we can go from field phenotyping through to gene identification, to validation, and then identification and characterization and field analysis. So this work was done through multiple teams, both led at Penn State by Jonathan. I've mentioned Paul Heinemann, who's doing who leads the ag engineering team. Molly Hanlon, who is the PI. Hannah Schneider, whose work I've just described on root angle. In Wisconsin, Sean and Natalia have done some great work in terms of assembling the germplasm and the GWAS analysis. At Georgia, we have Alex Bush and his team who've done the image analysis, and at Nottingham, we're part of the deep gene discovery pipeline. So I'd like to stop there, and I'd like to now possibly take questions at this point.
Yes. So there is a question in a chat. We can also open, if there are any other questions, you can raise your fingers and we can give the voice to any participants. But I will start with a question in a chat, which is the question is as follows: deficiency of nutrients, for example, nitrogen, phosphorus, potassium, is known to influence RSA development. Can field-based phenotyping approaches be used to map RSA under nutrient deficiency stress, which is seemingly difficult to impose in native soil compared to drought or hypersalinity?
It's a very good point. Obviously in many developed countries, where soil has been over-fertilized, you're indeed right. It's very difficult to find agricultural trial areas in which nitrate nutrient levels are sufficiently low to impose nutrient stress. And so it is a concern. To address this, we work in overseas. For example, we're doing work in Senegal where we don't suffer this problem. The soils are relatively nutrient poor, and therefore we're able to impose nutrient stress conditions. Jonathan likewise has done this, for example, in South Africa or in Arizona. So I think it's a matter of if you don't have access to obviously these conditions, securing them maybe in a different country, obviously that is extremely challenging practically, but there are also ways you can intervene to reduce nutrient levels. And I know at Penn State that they've got a program to reduce nitrate levels through various agronomic practices to deplete the soil of nutrients. So there are ways to actually prepare experimental plots which actually do fulfill these conditions without having to go take the steps of going abroad. But it's about having access to such facilities. Okay.
There is another question. Have you used high-resolution functional plant phenotyping pipelines?
Well, I'm going to talk about that in the next section, which is really going to talk about high-resolution imaging such as CT, in which we can obviously capture very very detailed anatomical features non-invasively. Clearly, within a field environment, it's extremely difficult to do so, and we have to adopt invasive methods. I know as part of the Roots program, the ARPA-E, there were other projects in parallel with ours that were looking at using techniques like CT to try and do non-invasive but high-resolution imaging in the field. I don't know where the advances they've made, but efforts are being made to achieve this in the field. But I think these are early days.
There is a question about root harvesting in a field, which is a mechanical process. So do you lose roots material? If yes, how much do you think this affects the results?
Oh, that's an excellent question. And of course, whenever you recover a root from soil, you're concerned that you're losing material. And obviously, if you use multiple methods, which are independent, for example, if you're using disruptive approaches, if you use shovelomics in parallel with, for example, soil coring, you can essentially use independent methods to try and assess this. But obviously, you do underestimate the amount of root material you're recovering. So it is a shortcoming of the approach.
Yeah, there is a question, a different perspective. You mentioned that most of the CO2 produced in the US could be captured by maize roots. This is a short year effect. So it's the question if it's a short year effect.
That's a very good point, and there's been a great deal of discussion about this. I should say that the science behind this was not our own. It was a US government scientist predicting that this could be the case. I am aware that carbon sequestration through such biological approaches, soil could effectively fill up. So I appreciate, this is not a panacea. It could be short. If you manage the soil poorly, you could in effect release the carbon back in rapidly, all the gains that you've achieved. So this really has to be about soil management, long-term soil management. Such crop breeding and agronomy approaches have to be used in parallel with that. But clearly there's likely to be an upper ceiling to this, and I agree. It could deliver short-term gains, but I suspect that if we think about how we've reassessed over the last 4 months through COVID, our energy use and our transportation usage, I suspect that we've reached peak oil, or we're close to it. And so this problem is likely to be relatively short-term as we move to more sustainable approaches. So it could provide a short-term solution, while clearly any alternative energy sources really are at the key of a sustainable future.
There's a question related to soil composition. So do composition of different soils influence the way phenotypic traits are mapped?
Absolutely. So one of our dirty secrets as a community are we love to phenotype in sand. Okay, nobody wants to. Let me just get rid of this very irritating Teams messaging system. Nobody wants to phenotype in clay because it's a nightmare to wash the roots and to then have post-analysis. So many of us choose to use very sandy soils. So that's part of the reason why we love Senegal. It has wonderfully sandy soils. Similarly, Jonathan in Arizona and South Africa. But clearly we have to look at multiple soil environments. And so as part of the Roots program, we have sites across the US which have very contrasting soils. And they are challenging. And I'm in awe at the work of Hannah and Molly and others who put in huge amounts of work to prepare the roots. So it is a hard work. But it's the right thing to do. We have to look under many different soil types. There are Phil Benfey's favorite quote. He tells me there are 20,000 different types of soil. It's true. There are. It's a truly monumental many types of soil. But as long as we I think analyze our root traits in a core set of 4 or 5, I think we can reach some understanding. And this is where this actually, I think, is a very important point. As plant biologists, root biologists tend to talk to root biologists. We don't really engage with soil scientists, and I think that's really to our detriment. And I applaud the work of several people who reach out to and work in parallel with soil scientists, because it's vital. If we're really going to understand how roots grow, develop, we have to work in conjunction with soil scientists. So this is one of the things that I would really encourage listeners to really embrace and cherish, because we've got so much to learn from soil science, which will make our science so much more relevant, and better.
So I would say we make one final questions. There are some more questions, but we could address those questions potentially at the end. I would suggest one final question, then we move to the next part. And at the end, when there is time, then we try to address as many questions as possible. So there is a question: what are the consequences on how to overcome the effect of over-fertilized or undernourished soil conditions while phenotyping root in a field for soil-borne root diseases? So yeah.
That's a very good point, and you're probably referring to, as you know, in regions which have poor soils, for example, in Africa, they suffer from awful problems from parasitic plants. The name of it has just gone out of my mind. And this is a product of the plants growing in low phosphate conditions, releasing strigalactones, which then stimulate the germination of striga. And it's true, this is actually a problem arising from access to nutrients. So if fields could be fertilized with, for example, phosphate, the striga problem would largely go away, or could be managed more effectively. And so it's true, soil nutrient treatments are vital for agronomy in many poor soil environments. And I think that breeding, we tend to over-focus on the importance of breeding. Clearly, it's a great way to intervene and improve yields, but this has to be done hand in hand with soil science, agronomy, and management. So it really is only one part of an integrated solution. Okay.
Yeah, thanks a lot. So I would suggest we go to the second part.
Right. Can everyone see my slide?
Yes.
Yep.
Fantastic. Thank you both. Right. So let's move on to controlled environment conditions. Now, as I explained, the field is a demanding and a challenging environment. And obviously, if we can simplify, that obviously is a huge benefit. And So what I'd like to do today is illustrate some of the solutions ourselves and others have come up with to image root traits in controlled environments. I'm going to talk about soil-free approaches. I'll talk about aeroponic approaches, rhizotrons, high throughput imaging of roots in pots, and finally I'll talk about some of the work that we're doing using X-ray micro-CT. But I could also, this is by no means an exhaustive list. There is some wonderful work done at ULIC on MRI, for example, and also, for example, in soil-free conditions with agar-based approaches, pioneered by Phil Benfey and others. So this is just meant to give you a taster of the various controlled environment approaches that can be adopted. Okay, so I'll start with a very simple system, which the concept of using this approach goes back a long way in plant science. Jonathan Lynch originally came up with such a paper method. This has been used by a number of different groups, and two colleagues at Nottingham have really seen the benefits of this. This is the pictures in the right-hand corner. You've got Darren Wells and John Atkinson, and they've developed a very nice high throughput method which uses this blue paper. So basically it's a hydroponic system. They're grown on this blue paper system. You can grow seedlings for up to 2 to 3 weeks. You're looking at the embryonic root system, and we offer this as a platform for researchers to come and use as part of EPPN that Roland described earlier. And this is a picture of, in the left, of András, who's come over from the Hungarian Academy of Science and as part of a transnational access project. In this, he was looking at nearly 500 wheat seedlings, different ecotypes, and he was taking images of these under different treatments and time points. As you can see, it's a high throughput approach. John and Darren have developed ways to very rapidly capture the image. The roots are grown behind a plastic screen, and then in parallel with computer scientists, they've developed some very nice semi-automated and now automated software. And this is the work of Mike Pound. So Mike has developed a very nice deep learning based software which called RootNav2, which builds on his original RootNav software, which takes those images, and within a matter of hours he can analyze 900 images, about 10 seconds an image, which illustrates the power of the deep learning approaches, and I'll come on to that a little later. So using these deep learning approaches, András was able to analyze the data much faster than he was able to capture it. And this is, I think this illustrates the incredible speed at which image analysis is really impacting crop phenotyping. If you want to grow plants in an aeroponic system, I would point you towards Louvain, which, sorry, here we go, I'll just move this. So University of Louvain, this is the platform which has been developed by Xavier Drey and colleagues. He's part of the EPPN network, and they have a fantastic system where they can grow hundreds of plants, and they can, unlike the previous system, which grows for the first up to the maximum 2 to 3 weeks, this can grow almost throughout the entire life cycle. And so this captures a very high resolution, the root architecture. And this is a very, very powerful approach, which obviously allows people to look at core architectural traits such as root branching densities and such like. Okay, so for some reason I'm stuck here. Okay, 1 second. Okay, I apologize. I'll just escape for a second. Okay, I'll stop sharing for now. for some reason. Okay. I can't seem to find my cursor. Roland, can you help?
I don't know.
I'll just try and stop share. I seem to have lost
Jennifer, any suggestions from your side?
Oh, here we go. 1 second. I'll see if I can get rid of this. Okay. 1 second. My cursor has just disappeared, and I can't see where I am.
Yeah, let me see if I can stop sharing.
That would be great.
Yeah, let's see if that fixes it, and then you can go, and then you can share again. Let me do that.
That would be great. Thank you.
Mm-hmm.
Ah, there we go.
Okay.
There we go. Thank you so much.
No worries.
Okay, so let's move on to the next system. And obviously, what I've talked about so far are soil-free systems. And obviously, this is one step removed from reality. They're very attractive systems to use. But if we want to study root behavior in soil, we obviously want to study it with this added complexity. And there have been some beautiful systems developed, for example, at Jülich. There are also some very exciting new facilities being developed at Gartenschleben, in which roots can be grown in these kind of automated systems. You can see here the roots are grown in these thin rhizotrons, and this allows you
Malcolm, sorry for interrupting. We don't see the slides right now, or at least I don't.
Right, okay. 1 second. Okay, let me, I will share screen again. There we go. Thank you for pointing that out. Okay. Can you see that now?
Perfect.
Yes.
Yep.
Perfect. Okay.
Thanks, Malcolm. That looks great.
So this slide, which Roland kindly shared with me of the system at Jülich, you can see here many it can run into hundreds. It can be expanded to many hundreds of rhizotrons, which are imaged this centralized imaging system. This is obviously a beautifully engineered system, and it's being used to analyze root systems at really very late stages of development, and can also image both root and shoot potentially. So this is an excellent system. It's been deployed in a number of facilities, and this includes Gartenschleben, for example. Many of us are using in parallel high throughput LenmaTech style platforms, in which we're obviously have conveyors in which plants are growing and moving, where their shoot canopies are analyzed. Work pioneered at Dijon, for example, as part of the Phenome, the French phenotyping network, is now uncovering not just the shoot canopy, but also the underlying root canopy. And so there are various clever ways in which the roots can be imaged in parallel to the shoot. So for example, the pots can be removed, the outer edge, and to uncover a transparent inner surface in which the roots can be imaged. And these are being used at a number of different facilities in France, for example, and others are widely adopting these. So it's a very exciting extension. At our own facility at Nottingham, we've tried to image roots in 3 and 4D using X-ray imaging. As I've mentioned, Jülich has also adopted a very similar approach, but they've used MRI imaging. We have 3 different systems at Nottingham: a small one for very small objects such as soil particles up to egg cup sized roots in soil. We have medium CT scanner, which is great for up to about 15 centimeter wide pots. And then we've got the Beast, this very large CT system, which I'll show you in a second. So just to explain about the image analysis challenge for CT, CT allows you to take you rotate the image 360 degrees and you capture a two-dimensional slice. And the idea is you take many hundreds of these, if not thousands, and you reconstruct them. And by identifying the object of interest, in this case a root, and reconstructing them from this set of two-dimensional stacks. Now, obviously, this is the image analysis challenge. Seeing, it's almost like the wood from the trees, the roots from the soil. And I show this because it's a problem which many of us, all the previous researchers and platforms I described also face. We all have to build phenotyping pipelines. Capturing the images is just the first step. As a biologist, I tend to over-focus on this first step, but I understand the frustration of my colleagues in computer science who point out that 90% of the work remains to be done. So capturing the imaging, the acquisition is just the first step. We have to recover the root material, we need to quantify it, we need to then develop data management approaches, and then obviously you do something useful with this, for example, modeling and simulation. So there's much more behind the initial capture. And I'll illustrate this in a few examples. So for example, the ability to reconstruct the roots, again, that's a huge challenge because the density, the X-ray density of soil and water and air, soil and roots are very, very similar. And therefore discriminating them is challenging. We initially started this work over a decade ago developing object tracking approaches. Stefan Meyerhofer, a joint student with myself and Tony Pridmore and Sasha Mooney, developed object tracking approaches, and many other things have happened since. Root One is a nice software which provided an interim solution, but I think, forgive me for saying, I think that the solution is going to be again through deep learning based approaches. And my colleagues in computer science recently had a breakthrough paper where they used deep learning, which I'll illustrate in a second. So manual segmentation of these images is painful. It takes, to capture an image may take a couple of hours, but to reconstruct it often can take days using even commercial software. And this can be very, very challenging. This is where I think these new imaging approaches are truly transformational. So this illustrates a paper. It originally came out in bioRxives. It's now at EEA. It came out recently, and it was led by Tony Pridmore and Mike Pound. And Reza, a postdoc in the team, has developed a very clever way to essentially obtain root images in effectively an automated fashion. And this is a game changer because it allows us to essentially remove the soil from the roots and to uncover a whole host and quantify a whole host of different important traits. So for example, all of these are just a few of the various traits that we could quantify as part of, for example, a breeding program. And we're using this approach in my own team to look at, for example, root adaptive responses. This is one root adaptive response that we characterize in collaboration with Jose Dinnerty's team, who's now at Stanford. And this illustrates, for example, when we peel away the soil, we can actually see the roots. On the left-hand side, you can see how the roots are branching all the way around when they're in contact with moist soil. But when you go down a biopore, the root architecture is branching patterns very different. This is called hydropatterning. And we've taken this further, and we've recently uncovered the molecular mechanism of this. And if you're interested, the molecular mechanism is described in the paper in Science a couple of years ago. And the cellular and developmental, the higher scale responses are described in a recent paper in Nature Plants. So the holy, I suppose the holy grail of controlled environment imaging would be to be able to follow a root throughout its life cycle. And that's something that we try to do at the Hounsfield using a high degree of automation and robotics. And so the Hounsfield facility is part of this European plant phenotyping network. The manager is Craig Sturrock. He also offers CT root phenotyping facilities as part of the EPPN Transnational Access System. This is a Lego model we use for science fairs, and you can see we've just taken the lid off, and you can see the underlying labs. You've got, for example, the small scanner, the medium scanner, and then you've got this large scanner which is attached to the greenhouse. And to bring the samples to the large CT scanner, we use robotics. We're able to grow 1 meter long columns, 25 centimeters wide. These are then placed within these large open, this lead-lined room. The sample is placed inside this room. This room contains about 30 tons of steel, of lead, to obviously protect the users. The sample is rotated, CT scans are taken, approximately 8,000 slices are taken. This is a speeded up example. And this allows us to scan repeatedly samples and to build up an image of the life cycle of a plant. And this image I show here illustrates the life cycle of a wheat plant. And as you can see, these are seminal roots that are initially growing in the early stages of wheat development. But once tillering happens, we see this amazing flush of additional tiller roots, which happen late on in season to help grain filling. So this really provides an insight into this approach, this non-invasive time-lapse imaging really, I think, is very powerful because it really uncovers how plant crops grow. There is a lot of root growth going on. When we think winter wheat is doing nothing in the field, in fact, it's doing a lot underground. And the depth of rooting is quite remarkable at these early stages. We've used these approaches, these time-lapse imaging approaches, to uncover entirely new structures. This is work we've done with Ikram Bililu in Kaust in Saudi Arabia, and we were looking at how date palm roots grow. And this illustrates how these are time-lapse image movie done on our mid-CT scanner. You can see this incredible level of resolution that we've been able to achieve through using this CT scanner. And we've discovered entirely new structures within the roots, which appear to play important roles in plant adaptation. So this is a very powerful technique with many benefits, like MRI. It's non-invasive and it's able to uncover very high resolution images. If you're interested in the work that we're doing, if you'd like to use some of the images for your teaching or your research presentations, we'd be delighted to share those. These are available on what we call the Hounsfield facility. We have a database called the Hidden Half, and this is a team effort. Many of us, past and present members of the teams, have contributed to scan over 30 different species, and we're adding to this on a weekly basis. You've got anatomical data, you've got architectural data. This was possible through a European Research Council grant awarded to our team, which largely funded the team shown. This is a multidisciplinary team made up of mathematicians, engineers, computer scientists, statisticians, crop breeders, as well as plant biologists. So it's called a Future Roots team, and it was an amazing thing to be able to lead. So just to summarize, you can see there are many different approaches which can be used to perform root phenotyping in controlled environments. As mentioned before by Roland, there are about 31 facilities across Europe in which you can apply for transnational access. in terms of phenotyping, 13 of these are focused on root phenotyping. So please go to our website. There are some brilliant facilities. For example, I've mentioned Dijon. They have the high throughput system. And just to reiterate, the final call for this is in June, this month. The call's gone out. And so I would strongly recommend you apply, not only from, I should point out that 20% of the budget can be allocated to non-European transnational access applications. So we've welcomed people from South and North America and Australia to the Hounsfield. And so I'll leave it there, and I'm happy to take questions before my last section.
There are a few questions. The question, how much can we predict tolerant genotype based on the results of root phenotyping, say root biomass, during early vegetative, say seedling stage under drought stress?
That's a very good question. And I know that there is a degree of skepticism about early seedling traits. But if you imagine, early seedling traits really are the path, as my CT scan of the wheat seminal roots illustrates, these are the pathfinders. These are the deep rooting pathfinders, which play a very important role if, for example, there is drought applied. So, for example, if we apply drought, we see a very significant reduction in tiller root formation in wheat. Jose Dinanese has reported similar behaviors in other cereal crops. So this illustrates the importance of seedling roots in contributing to crop performance when adventitious roots may be inhibited in the upper soil profiles. So I actually think that the early seedling phenotypes are probably underestimated of their importance to crop performance.
There's a question about data. Are the data from the Moiety Species Scan available through a public website?
Right. In terms of the website, I think you can download the movies, the images. In terms of the actual data, I'll be honest with you, the CT data is a nightmare. We have a data storage of 180 terabytes, which is full of CT data. I would not recommend moving it, downloading that, because that could take a long, long time. So I think this is actually, you've touched on a very, very important point, which is one of the challenges we still face is moving image data. And so all our storage for the Hounsfield is on site because of the time it takes to move data down to remote sites. So I think this is we're exploring the whole issue of compression of image data, and obviously you don't want to lose, and how much you lose. I think this could be a very interesting area which could really benefit the community in terms of accessing these data sets. If you're interested in particular data sets, then we'd be very happy to share them, but we'd probably have to send it you on a hard drive. That's probably the most efficient way of moving such large data volumes. So yeah, CT data like MRI, it's very large. That is one of the, I guess, the downsides in terms of data management.
So I would say that there is also one question from the previous session. I would say the last question now, and then we would move on, looking also at the time, we move on to the last session. And at the end, we can potentially keep the session with questions as open as long as people are interested and as long as you are still able and willing to
yeah.
questions after already quite some time progressed. So the question deeper roots provide a benefit for drought resistance. The question is whether and how it affects nutrient uptake, like nutrient and phosphorus.
I mean, that's an excellent question. And it would be wrong of me to, I mean, deeper rooting, obviously, as I've talked, provides benefits in terms of accessing deeper water sources. It also allows the root more time to capture water-soluble nutrients, such as nitrate. That's actually the reason why we suspect that the deeper rooting maize variety I showed of Hannah Schneider's work actually is more nitrogen use efficient, because it's got longer down the soil profile to capture that nitrogen that's moving with the water. You're absolutely right, though, for nutrients such as phosphate, which accumulate in the upper soil, that is not going to be benefited. So developing root systems which are able to forage in the topsoils but also deeper is the obvious solution. And this is a really interesting question. It's a bit like life. Life is complex and simplifying it is naive. Modeling allows us to look at this complexity. And again, functional structural plant models such as OpenSimRoot, developed by Johannes Postma and Jonathan and others, is very important to simulate root architectures and to try and identify the optimal root architecture for a given crop. So I think model-driven phenotyping is really an area that is going to be very, very important in the future. So rather than do large-scale trials, we may have a far more targeted approach driven through modeling. I think that's going to be far more important, yeah, and could identify the root phenotypes which would address your question.
So should we move to the next part? Welcome.
Great. So I'll try and be quick. This is actually shorter than the others. So new innovations. I'd like to, a lot of what I've talked about today has been about redesigning root architecture, about how architectural traits contribute to function. But as I touched on several times, anatomical traits are also important. And this picture is a very nice example from one of Jonathan's papers, which illustrates parenchyma. This is the airspace where cortical cells undergo programmed cell death, and they you see this in rice likewise, in which the benefit of this is you actually reduce the amount of respiration carbon a tissue requires, and you can reallocate this for other purposes. So the ability to phenotype at the anatomical scale could be extremely important for looking at carbon efficiency, as I've illustrated with the xylem, water use efficiency. So, but obviously this has been challenging because it's not been possible. And I think a real game changer for this whole process has got to be the development of techniques like laser ablation tomography. And Jonathan Lynch requires real credit for this. So he was one of the co-inventors, developing this together with Ben, has a spin-out company at Penn State, and they operate a laser ablation tomography. In Jonathan's case, they have several instruments in their laboratory in which they phenotype samples obtained from the field, and they can obtain various traits. Now, obviously, the ability to image the roots is, again, to illustrate step one. And the subsequent quantification of these traits is hugely challenging. And various semi-automated to manual segmentation approaches have been used in the past. And as part of the Roots program, Eze and Tony Pridmore from Nottingham have been developing deep learning approaches to capture this automatically. And they've achieved quite remarkable results. They've been able to match, if not improve, manual segmentation approaches, which take many fold times more time to actually acquire the analyzed data. And these, I've just shown you a few examples of how maize lat images obtained have been analyzed by ESE software, and he's able to identify a whole host of different features, such as xylem vessels, for example, parenchyma, cortical cell number, etc. So this is, I think, an extremely welcome and exciting development funded through this ARPA-E program. One of the challenges is really image quality, and improvements in lat imaging is something that Jonathan, who's based at Penn State, but also has held a joint post at Nottingham, has been very keen on. And so working with colleagues, John Atkinson and Darren Wells, and Ben at Penn State, we've acquired what we call LAT 2.0. And this uses an athermal laser, so a more advanced laser, in which samples are then sectioned. And we're using these more advanced lasers in combination with improved optics, for example, using microscopes rather than macro lenses on cameras. We're able to achieve extraordinary levels of resolution and uncover subcellular features. So as is shown here, you can actually see the Casparian strip that's dividing the outer cortex in the inner vascular tissues. In this case, this is a pearl millet root, which we've sampled from Senegal. And we're so excited about the opportunities that this types of technology offers. And so we're using this LAT system, LAT 2.0 system, in a Royal Society funded project that's joint between colleagues in Senegal, France, Laurent Laplace at IRD, at Nottingham and Penn State. And this project is using performing field trials in Senegal, where you can see it has wonderful sandy soils, perfect for obtaining the root samples. These are then, we undergo a process which we call anatomics. So it's the omics of anatomy. And we're able to take the samples, prepare them, and then section them using LAT. And once we image them using LAT and then employ deep learning algorithms, we're able to then plug them into genomic data sets by performing this analysis on genome-wide association panels, as I described before for maize. We're able to then identify key loci which control traits within the root, the pearl millet anatomics data sets. And by doing so, we can drop onto potential candidate genes to improve drought tolerance mechanisms. Just to show that this is possible, some work that Rahul Basole has been doing. He's a BBSRC Discovery Fellow. He's been working closely with Hannah and Molly and Jonathan and Sean Kepler's team. This illustrates how GWAS data from, in this case, a maize diversity panel has been used to identify underlying genes which control a parenchyma trait. So these are identifying a whole host of genes which you'd associate with parenchyma, for example, hypoxia, programmed cell death, reactive oxygen signaling, ethylene signaling. And we're very excited by the candidate genes which this new form of omics, anatomics, is uncovering. So just to sum up today, what are the key themes? Well, I think that root phenotyping techniques like LAT and CT, MRI and others are helping uncover the hidden half of crop performance. I hope what's come across very clearly is multidisciplinary approaches are really key to uncovering new insights into root growth and development. Active engagement and uptake of approaches from these disciplines and these technologies is critical. We have to come out of our comfort zone. We have to reach across to other communities. if we're really going to take our science forward. And clearly, it's not just on us, implicit on us to actually do this, but also international initiatives such as EPPN 2020, Emphasis, IPPN, and others are really helping us achieve this goal through pooling tools and resources, for example. If I may, I just want to give a quick plug for a meeting that's taking place next summer at Nottingham, we hope. It was meant to be this year, but due to COVID, it had to be relocated. We made a pragmatic decision to move it at the same week as the International Society of Root Research meeting in Missouri. This was done in conjunction with Bob Sharp and other co-organizers of the ISSR, and Michelle Watt, we particularly thank for being open-minded and supporting this. ISSR is running in North America the same week. We're running a session, the meeting at Nottingham. This week we're going to share plenary sessions. We're going to share technology sessions, one of which is funded by the IPPN, and we're going to be sharing various theme sessions. So talks are going to be beamed across. The afternoon sessions in the UK are beamed across and shared as morning sessions in the US, and shared poster sessions. So it's a totally new way of doing science that's really designed to allow you to maybe stay within a continent or to travel surface rather than air and participate either online or in person at these events. And we're really excited about delivering this new type of format. I'd like to just finish there, and I'm really happy to take questions.
Thanks a lot, Malcolm. You've talked now for nearly 91.5 So to give you also some opportunity to catch the breath, there is someone from the group, from the participants, advertising about a special issue, MDPI special issue on mechanism of root development, shifting from 2D to 3D and 4D analysis of molecular resolution at cellular level. I think that's a relevant information for any participants, so papers can be submitted. And I think, Malcolm, you could very briefly catch some breath before we start with the next questions. And so the first one would be already, can you monitor nutrients movement into root and shoot?
That's a really interesting question. I know that there are a number of people who are trying to use various sensors to achieve that goal. So researchers in Manchester, for example, are developing approaches to sense nutrients by inserting sheets, which essentially will detect key nutrients. So there's a lot of activity in terms of sensing nutrients like nitrate, non-invasively monitoring them, but obviously they represent real challenges. So, for example, ourselves, we're looking at developing biosensors for hormones, but again, these tend to be relatively invasive. And so I think it's an area that really requires development and reaching out to biophysicists and biophotonics experts. So I think this is a really exciting area that requires much more funding and development. So thank you, that's an excellent question.
So another question, one good thing in soil-free media is that the roots don't suffer damage, and it'll be easy to use them for downstream studies. How much damage do roots suffer in native soil? How would it interfere in studying something like root cell biology, for example?
That's a really, yeah. Obviously, if you grow them in aeroponics or blue paper or those soil-free systems, it's a lot easier to recover the roots. I think people think it's a lot more difficult than it is. Our own lab is doing a lot of work, for example, looking at soil compaction, and recovering roots from highly compacted soil is challenging, but possible. So one of the researchers in our team, Bipin Pandey, has gone to extraordinary lengths to recover root material, and we can get beautiful confocal images from them. So it takes care and effort. It's not high throughput, I admit. So this is for more detailed studies. But of course, there's a degree of loss which you have to obviously assess and take into account. But if I may make a plea, I think we should really all get mud underneath our fingernails. I lived for the first 20 years of my career, I believed roots grew in agar plates. It's only in the last 10 years working with soil scientists like Sashi Mooney and breeders such as Jonathan that I really start to appreciate the importance of working in the real material. And it's challenging, but it also is very rewarding. And you discover entirely new mechanistic adaptive mechanisms, and it's a black box, but there are so many things, cool things to discover which haven't been so far. So for a young PI, I would truly encourage you to think about, delving into the hidden half. There's much more to discover.
There is an ambitious question. Have you tried millet roots with 3D resolution for 40,000 cells?
That's an interesting question because, clearly the deep learning algorithms that we've developed as part of the Roots program so far is focused very much on analyzing two-dimensional sections, okay? But in principle, these deep learning algorithms can be expanded to three-dimensional. Because it's a destructive method, it can't go to four-dimensional, obviously. But it's possible. It's not high throughput, but it could be with enough time and effort. So ESE is making real progress on the 3D deep learning algorithms, but obviously it's a lot more challenging than 2D. There is commercial software out there that can take LAT data sets and generate some beautiful graphics. But again, these are lovely images, but quantifying the features is another challenge. And so I think this is where the 3D deep learning algorithms will really be instrumental in achieving that ambition.
So there is a response from the person asking the question that there is apparently such a map with 3D.
I'm sorry, that's my telephone. Please ignore it. It'll go off in a second. Carry on.
There is also a next question. What would be an effective methodology to get quite good fresh mass of root washed out while working in a field? So, methodology for, yeah.
Many people use, for example, soil coring, and they're a variable efficiencies. I mean, I think this whole space requires huge, real investment, not only by plant biologists, but as illustrated in the deeper project. Getting ag engineering experts involved really opens up new opportunities and solutions. So I think that this really illustrates one of the challenges. We need to develop more efficient ways of recovering root material. One way to do that is to obviously develop larger soil cores. So we've been doing that. We've been, for example, soil cores traditionally are a few centimeters wide and might go down a meter. We're developing soil cores, as are the Australians, of 20, 30 centimeters wide. This is actually quite challenging engineering wise, but we bring them back to the Hounsfield and we scan them. So it's possible. It's possible to do this. And we're getting extraordinary images back out of these field structured soils. We're discovering about, the impact of root growth down cracks and how difficult roots are able to penetrate subsoils. So I think it's a very important question, and we have to be imaginative in the way we address it. And the example I've given you about coring it, using, in this case, we used a commercial coring company, and we brought back the cores and used the large CT scanner. And this work was published recently. Have a look for it. It's led by Sasha Mooney, and it uncovered amazing images. Basically, roots beneath the plough pan for wheat tend to largely go down the biopores. They really struggle to get into the bulk soil below the plough pan, and so the insights were very, I think, interesting.
There is a question with respect to climate change about approaches to basically reduce CO2, so carbon sequestration in soils, if there are any approaches that could be used for that purpose.
Sorry, that's my co-worker, my cat. It's a very noisy cat. Clearly, the deeper program advocates using breeding approaches, and that is obviously, I think, a highly effective way to do that. But clearly, there are other approaches which could be used in terms of carbon capture, which are obviously outside the realms of which are more engineering focused and outside the realms of plant breeding. So I think a crop-based solution is what we could offer. Obviously, looking at how the microbiome releases carbon, developing ways through soil management or intervention approaches to reduce release of CO2 from the soil microbiome could also be another way. I mean, agronomic practices are very important. When we plow the soil, we release a huge amount of water and release carbon stores. So no-till and min-till approaches are very, very effective at reducing this release of carbon and loss of moisture. So even without new crops, we could do, and farmers are doing it, changing tillage practices can really deliver big benefits. And as we talked about earlier, really, these are the short-term solutions we could deliver real benefits. Obviously, developing more sustainable tillage practices are attractive and should be welcomed. So again, it illustrates plant breeding is just one part of an integrated solution.
There is a question, interesting question. Do you think it's possible to study the impact of plant pathogen interaction on root growth development using in-soil phenotyping techniques?
Well, we've been very active in using the CT to look at the impact of pathogens like Rhizoctonia. We've also worked with colleagues to look at the impact on roots of striga on root architecture. In this case, we used CT, the mid-size CT, and we obtained really extraordinary images. And so I think a lot can be done in controlled environment for these types of analyses. We don't have to go to the pain of doing this in field, which, as I've illustrated, provides a lot of challenges to do this non-invasively. So I would say these type of studies, a lot can be learnt from controlled environment studies. So please have a look at some of that work that's been done with colleagues in Sheffield Julie who's a Striger expert. She's done some lovely work in that area, and we've worked with plant pathologists as well with looking at the effect of Rhizoctonia. So I would say for people who work on plant pathogens, really think about applying to the EPPN and coming to one of the platforms and studying your favorite pathogen using these non-invasive approaches.
So there is a question also about what would be a powerful method for measurement of mixed cropping. I assume that it's more related to roots, so that it's not specified in detail, but in the end you could do for roots and shoots, and I suppose it's about root interaction in mixed cropping.
Yeah, I think this is really interesting. I mean, I'm conscious that many of today's participants are from the US, obviously being held through Nebraska, and you know, they advocate the three sisters. So you've got beans, maize, and squash, and that was a vital system for the Indian population and I think, you know, people such as Larry York, who I noticed was attending, has done some lovely work on this topic area. We have developed software approaches to actually address this. So we've developed segmentation approaches to actually follow multi-species or even multiple individuals within a single species, because, you know, obviously looking at monoculture is important. I think the whole issue of studying roots as individual plants is naive, and the question, I think, reflects that, in the sense that, you know, we should be really looking at plants as part of a population. And so obviously developing imaging approaches either as monocultures or intercrops, I think are very, very interesting. And I think that we do have the software tools now to be able to segment multiple plant systems within a single scan. And it will be very interesting to look at questions of competition and interaction within a real soil environment.
So I think we went through most of the questions. So unless there are very urgent questions that would be posted now, I would like to thank you, Malcolm, for the really inspiring talk and for all your patience in addressing all those many questions. And right now there is a number of people thanking you as well for the very inspiring talk. So thanks again.
Well, thank you very much for all your questions and your. I really appreciate having this opportunity, and I'm sorry it's gone over a bit, but maybe that reflects my enthusiasm and hopefully the interest of the audience. But thank you everybody for your support.
Great, thank you, Malcolm. Thanks everyone. Have a great weekend.
All right, thank you, Jennifer. Thank you, Roland. All the very best.
Sure thing.
Thanks, Malcolm.